Is it visible to all of you?
Yes, sir. Yes, sir.
Okay. So today I will be talking on the advances in biological wastewater treatment processes. As we know that biological wastewater treatment processes are used as the secondary treatment processes in the sewage treatment, because normally traditionally we know that after primary treatment the secondary treatment processes are used. So first we will see the introduction of this, then advanced biological treatment methods which are available, and then we will see the case studies and then the conclusion. So first of all, biological treatment, why it is required and why it is done. So biological treatment generally, if first of all we should know the wastewater. What is wastewater characteristic? So as we know that
Okay, fine है. आवाज आ रही है सर?
So yes, okay. So the biological treatment, first of all, we should know why it is used. Like wastewater, we know that the wastewater which is generated, whether it is generated from domestic or it is in the industrial wastewater, having some organic matter into it. Okay. So as you know, how the organic matter is generally measured by the environmental movement. Organic matter, you know, that is many of you are from the chemistry background, and some are from civil engineering, some are from other chemical, some of other background also. So the mixture of participants are there. So first I will tell you what is the characteristic of the wastewater which is generated. Now, as you know that from the domestic wastewater which is generated, what it is having, whether we can directly release into the streams or not. First of all, that we should know. Okay, whether we should directly release into the streams or not, like rivers or some other water bodies, oceans. And what happen if we release this wastewater directly into the river or oceans? I think this is one question. Just generally, I ask the student, what will happen if we release our wastewater or the sewage directly into the river? What will happen? If any answer can give, I will be happy if you give the answer. Yes, actually.
BOD will decrease.
BOD will decrease. Any other participant? No. What will happen if you directly release your effluent or domestic wastewater into the river or ocean? Directly, not giving any treatment. Basically, environmental engineer should know. Anyone? I think chat section. Anyone? Yes.
Sure.
Yes.
I think for anaerobic digestion, I think combined oxygen is required or not?
Because I think without oxygen, anything, nothing can be decomposed.
My question is, what will happen if we just if we release our effluent into the river? कोई treatment नहीं करें, no treatment is done and just release into the river. What will happen?
So it is going to damage this aquatic life.
DO decreases.
DO decreases, yes. Yes. So
And it cause damage to the
yeah.
this thing, aquatic life.
Yes, it will damage the aquatic. So what is happening that if we directly discharge our wastewater into the river, wastewater contains what? Wastewater contains organic matter. Organic matter is whatever the, like you can say the carbohydrate is organic matter, the proteins is organic matter, fats is organic matter. These are all organic matter. Okay. So all these are organic matter, and if this organic matter, if you release into the river or any ocean body, what will happen? That the bacteria they start degrading it. Okay, the organic matter that is released into the wastewater, the bacteria get degrading it. So normally in the river or ocean, dissolved oxygen is there. All of you know the dissolved oxygen is there in the river or the any water body, and that dissolved oxygen which is there, like it is around 8 to 9 milligram per liter is there, depending upon the temperature, depending upon the altitude, depending on this dissolved oxygen may be more or less. Okay, but we take, let us say it is around 8 milligram per liter, generally around 8 to 9 milligram per liter dissolved oxygen is there. And our this aquatic species like fishes and all other species, they take this dissolved oxygen for their respiration. Like we are doing, what we are doing, we are taking the oxygen from the atmosphere and we are doing the respiration is done. Okay, so this aquatic species, they are also taking the dissolved oxygen from the rivers. What is the dissolved oxygen there? They take it and they are surviving into it. Okay. What happens if you discharge the wastewater? Other than the aquatic species, there is many bacterial species are there. So bacteria, aerobic bacteria, what they are doing? They start degrading it, whatever the organic matter is there. Okay, they start degrading it, and as they degrade, you can see here, as this aerobic bacteria will degrade these organic contaminants, okay, in the presence of oxygen. The aerobic bacteria, they require oxygen. So oxygen plus organic matter, so this is the reaction you should note down. Organic matter plus oxygen gives CO2 plus H2O plus energy they will get. The bacteria will get energy. They will take the oxygen, organic matter as a food. That is the food for them. We are also taking organic matter as a food. हम लोग क्या लेते हैं? What we are taking? We are taking the rice, and they are organic things. We are taking the all the vegetables. They are organic. All the ghee, butter, they are all organic. So why we are taking it? Because it gives us energy. Okay, all living organisms, they require food for getting the energy. So like that, bacteria is also getting the organic. So organic from where they are getting? From the waste. In the waste, the organic matter is there. So the bacteria is taking that organic matter. They utilize the oxygen. They take the oxygen because all the respiration reactions happens in the in the presence of oxygen. The respiration, if you are talking, so organic matter plus oxygen gives water and carbon dioxide. Carbon dioxide is released, and in that way, bacteria, they reproduce also by binary fission. Okay, like when they get the food, the bacteria will be reproducing by binary fission, like from 1 to 2, 2 to 4, 4 to 8, like that they are increasing. Okay, so what is happening that they are consuming the organic matter and oxygen, and they are giving water, carbon dioxide, okay, and they are getting as a food. So what will happen? That waste they will consume, but what else they are consuming? They are consuming oxygen also. From where they are consuming the oxygen? They are consuming from the river. जो भी dissolved oxygen है, whatever the dissolved oxygen is there in the river, they are consuming it with the organic matter. So that is the respiration reaction for getting the energy. Organic matter plus oxygen. We know, we have learnt in our schools also, respiration reaction. C6H12O6, that is the glucose, that is also organic matter. C6H12O6 plus O2 gives CO2 plus H2O plus energy. Okay. So bacteria is doing that only. Same thing it is doing for getting the energy. So it is consuming organic matter, but it is consuming oxygen also with that. Okay. So what will happen to the dissolved oxygen in the river? From 8 milligram per liter, what will happen? It will decrease. From 8 it will be 7, then it will be 6, then 5, 4, 3. More organic matter you will dump into the river. What will happen? This dissolved oxygen will decrease rapidly. Okay, from 8 to 7, 6, 5, 4, 3, 2, 1 and 0. Okay, what happened? After 2, if it decrease like after 4 only, below 4, 4, 3, very less oxygen will be there for the aquatic species. If the fishes or the aquatic species, any aquatic species, they cannot survive at this low DO. Why? Because why they cannot survive? Because what we have done, we have released the organic contaminants into the water, and due to releasing this organic contaminants and oxygen, the oxygen has become less, and due to becoming less, the fish will die. So that is due to our the human beings. They are discharging all this wastewater which is generating from sewage, which is generating from industries to the river or ocean. So it is our responsibility to treat it. So there is one method of treating it, biological treatment, because it is a very efficient method which do not require any chemicals. So it is a nature way of degradation. It is the nature way. Okay, here we are not using other way, we are using the nature way. So this is the aerobic treatment principle that so what we are doing? The same thing that is going to happen in the river, that we are going to do in our reactor. Okay, what is happening, what is going to happen in the river, we do in the reactor. Now this organic contaminants, how we measure it? How you can measure this organic contaminant? How much amount of organic contaminant is there? How we can measure it? If anyone knows how to measure this organic contaminant. Anyone knowing how to measure this organic contaminant?
Levels of BOD?
Yes. So we are using the BOD method, biochemical oxygen demand, or we can measure it by chemical oxygen demand, that is COD. What is this? How we are? Yes, Mamta Sharma, BOD. So we know that biochemical oxygen demand is nothing but we are doing one practical in the lab where we are using we are taking the wastewater into and we are diluting it, and what we are doing, we are finding out initial DO and final DO in the lab, and the calculation is like that: initial DO minus final DO divided by dilution gives you BOD. That's simple. Initial DO minus final DO gives you BOD. Which is more, initial DO or final DO? And we keep it for 5 days. Okay. What we do, one bottle is there, we fill it with the wastewater, diluted wastewater. We keep it for 5 days at 20 degrees Celsius, and after 5 days we measure the DO. So first we are finding out the DO, initial DO, and after 5 days finding out the final DO. Which will be more, initial DO or final DO?
Initial DO.
Initial DO is more, final DO is less. Why the final DO is less? Do you know? Why the final DO is less?
Because these microorganisms are consuming that oxygen from this water.
Yes.
To decompose the organic matter.
Yes. So in 5 days, what is happening? The same reaction that I have told you just now. Organic matter plus oxygen gives CO2. So microorganism, what they consume the organic matter in the wastewater in that bottle, and they consume the oxygen. So initial DO minus final DO gives you the BOD. Now how it indirectly gives you organic matter, that is final DO, you know. If final DO, let us say the initial DO is 8 milligram per liter and final DO is in one, it is 5 milligram per liter, in other wastewater it is 2 milligram per liter. Which has more organic matter? First one or second one? Let us say initial DO is 8 milligram per liter. In one bottle, the final DO is 5 milligram per liter, and in another bottle, the final DO is 3 milligram per liter. Which one has a higher organic content? First bottle or second bottle?
The second bottle.
Yes. Second bottle has the higher organic content. Why? Because in first bottle from 8 it becomes 5. In second bottle from 8 the oxygen level becomes 3. Means the bacteria has done more consumption in second bottle. It means indirectly that there is more organic matter in the second bottle, therefore the bacteria has done more consumption. And you know, DO is initial minus final. So in first bottle it is 8 minus 5, that is 3. In second bottle 8 minus 3 is equal to 5. So 5 divided by dilution, let us say if you have done that dilution 100 times, then the BOD is 500 milligram in second, and in first it is 300 milligram per liter is in first. So fine. So BOD indirectly what it is giving? It is giving you the organic matter present in the wastewater. BOD is giving the organic matter. And how we are doing this test? We are utilizing the bacteria in the bottle to do the test to find out the organic matter. COD is the same. COD is not, we are not utilizing the bacteria, but we will be utilizing the chemical like potassium dichromate, K2Cr2O7, which is a very high oxidizing agent. We are oxidizing this wastewater, and we are finding out how much K2Cr2O7 is consumed. More K2Cr2O7 is consumed, it means the COD is more, means it has more organic matter. So this BOD and COD test is to determine the organic matter in the wastewater. Okay. So we come to this. So this organic matter, so BOD means organic matter. Just you should be BOD means organic matter. So this organic matter plus oxygen, when it combines, it gives water and carbon dioxide. Okay. Anaerobic treatment. In anaerobic treatment, some bacteria is there which do not require oxygen. Okay, they can directly convert the organic contaminants into carbon dioxide, and they have generate methane also. Okay, in addition to carbon dioxide, this anaerobic bacteria will generate methane also. So anaerobic bacteria, but they are the slower one. Okay, they do it very slowly. So like if you discharge any wastewater into the river, what will happen? First, it will be very fast. Okay, 8 to 7, 7 to 6. The oxygen level will decrease fast. 8 to 7, 6, 5, 4. But if you have very high quantity of wastewater into the river, it will come to 8, 7, 6, 5, 4, 3, 2, 1, 0. After becoming 0, whether the degradation will stop or not. After the DO becomes 0 in the river, whether the degradation will stop or not.
Yes, sir.
It will not stop. It will be going after 0 also. And that is by anaerobic bacteria. Okay. But you know why we don't want the anaerobic condition to be prevailed? Because then this reaction is very slow. Anaerobic, it takes days to degrade. Okay, and many gases will also involved. That you may have noticed in the nala, and where very much high amount of organic matter if you put, the DO becomes 0, and then anaerobic treatment is anaerobic digestion starts, and in anaerobic degradation, these gases will be released. Various noxious gases will be released. Okay, so this anaerobic bacteria works when there is no oxygen. Okay, but we don't want this condition to come in our water system, anaerobic condition. We are utilizing this anaerobic things in our reactors, that we will see afterwards. We are utilizing it in our reactors, anaerobic reactors. Okay, so now we will go further. So now the biological process where we are using the aerobic reactors, they are called activated sludge process, aerated lagoons, waste stabilization pond, that is polishing ponds, then trickling filters are there, and anaerobic, they are anaerobic digestion, and other systems are there. So first of all, we see the aerobic system, system that is called activated sludge system or activated sludge process. So what we are doing, we are using the same reaction which we have done, which is the nature's way of cleaning, into a tank. Here what we are doing, so we are doing that thing in this tank. So what we are doing, this effluent, the sewage effluent is coming into the tank, and we are aerating it. Okay, we are aerating it. Why aeration is done? Because what is happening in the degradation, your main thing is the DO. From 7 it will be 6, 5, 4, 3, the DO will decrease. If you maintain the DO, what will happen? If you give oxygen, what will the bacteria will do? It will degrade all the organic matter. The main constraint is the oxygen. But if you give the oxygen, what will the same reaction will be happening in no time. Okay, if you give the air, because bacteria, they requires only oxygen and food, and they will multiply like anything. Okay, 2 to 4 fold. I have told you that is the binary fission by which they are multiplying and they are degrading it. Their quantity increases and they are degrading it. Degrading means they are taking as a food what we are using. Okay, what for food we are consuming the food. So they are consuming the waste. They are consuming the waste. Only problem in the river was what? In the river there is a limited quantity of oxygen that is to be utilized. Okay, but in aeration tank we are giving oxygen to them. Okay, treat it. We are giving the oxygen to the bacteria so that they will treat it. So here that treatment will be done, means the same reaction will be happening. Okay, this reaction will be happening. Organic matter plus oxygen, that gives water and carbon dioxide. The same reaction is happening. Okay. Now this reaction will happen and everything will go into the secondary sedimentation tank. Okay. And in the secondary sedimentation tank, what is happening? Whatever the bacteria which is generated here, it will be in the mixed form, because due to aeration there is a mixing also happening. So everything is in suspension form, suspended. Everything is in suspension. Okay. Maybe you have seen the, many of you are environmental engineers or civil engineers, you have seen the sewage treatment plant and the aeration tank. So there the diffusers are there from where the air will be going out. So here the air will be coming out. You can see here a pipe will be there, pipe is coming here, and through the diffusers the airs will be coming out. Like same thing that we have seen in the aquarium. If you have not seen the effluent treatment plant, you have seen the aquarium. So there how the diffusers are happening, that same thing is here. Now everything is in mixed condition, and then all thing will go into the secondary clarifier here. And in secondary clarifier, what is happening? All the bacterial sludge will settle down at the bottom, and on the above you will get a secondary clarifier effluent. Now the BOD of this will be less. The organic content of this secondary effluent is very less. Where the BOD has gone? Initial BOD is, let us say, 400 milligram per liter, and the final BOD is 30 milligram per liter. So where the BOD has gone? It has gone into, the BOD is converted to CO2 and H2. Organic matter plus oxygen gives CO2 and H2O. Everything is converted to CO2 and H2O. Okay. So your BOD from 400, when it comes out, it becomes 30. So 30 is the limit that we can discharge to the river. So that is by law also. Okay. Our pollution control board, if you discharge your wastewater more than 30 milligram per liter BOD, it is offense. You cannot do it. By the law, you cannot discharge your wastewater which is having a low BOD. Okay. So into the river water or any water body. So that is coming out. So from 400 it becomes 30. Now what we are doing here, whatever the biomass which is settled down, we are recycling it back into the system. That is called return activated sludge. We are recycling this back into the system. Do you know why? Why we are doing this? Why we are recycling it back into the system? The sludge which is generated, or the biomass, the bacteria, we are recycling it. If anyone knows why we are recycling the sludge or returning the sludge. Okay, so one candidate has written, Mamta Sharma, more microbes will degrade more. Yes, that will be true. But we have to be very careful in recycling also. Can you return more thing? All the sludge? Can you return all the sludge or some of the sludge? We cannot return all the sludge back. We cannot return all the sludge back. Some portion of it is returning back. Why we are doing it? Some portion, like I'm giving this to our to my students also. Like let us say in one room we are there. Okay, so let us say we are like a bacteria, and the food has come. We are in the aeration tank and the food has come. So what we will do? We'll eat the food. Okay. But let us say we are 3 persons and the food is more. So what will happen? we'll eat the food, and then there's a left out food that will go outside. So left out food is more BOD. Okay, we'll eat 3. So or we will 5 is there. But what we are doing, we are coming, this bacteria is coming here also. But if we recycle more, like in 3, 2 more will come. So 5 will be there, or 10 persons will be there. They eat more food. So more BOD removal will be there. Okay. But we have to see that we are consuming oxygen also. Okay. So oxygen content must be there, and oxygen from where we are providing? From the outside we are providing air, and air is not a free. What we are providing air is not a free, because it is compressed air. That is, our compressors are there, okay, or blowers are there, which are providing the air. So you know, in ETP, effluent treatment plant or sewage treatment plant, mainly the more cost is involved. That is due to this aeration, this air which we are providing, the compressor and the blowers which are running day and night, that is consuming more electricity. So if we return more activated surge or more bacteria, let us say we are in our I have 4 people are in one room. Okay, now what will happen, that more people, if we allow to come, then though no food is left, but they will consume oxygen, because more people are there, they will consume more oxygen. So why to have? So we have to balance it. Food and people who are there, that we have to balance. We don't want that people will be less and food will be wasted. We don't want that people will be more and they will consume oxygen. Okay, so both the things we have to balance. So we have to return the amount that is required only. Okay, so this is activated sludge system, and if you see here, this is the clarifier sedimentation tank. Okay, now, and from here we I think this is not clear, no problem. We come to the design parameters of commonly used sludge. I think I will show this. So this is okay. So this is the secondary clarifier, and this is the aeration tank. You see, the primary effluent is coming from here, and that return sludge is coming here. You see, that is so much dark due to the sludge, that is the biomass which is coming back in the system. So this aeration tank is there. You see, there are so many bubbles is there. So everything is in mixed condition, and primary effluent is there, and then the return sludge is there. So this is the aeration tank, and this is the secondary clarifier. Now next is the design parameters for commonly used activated sludge. As we know, there is a mean cell residence time, and there is a food to biomass ratio that we have to maintain. Okay. Now food to biomass ratio, that I was telling, that food is what? Food is BOD. BOD is the food, that is organic matter. BOD is what? Organic matter, that is the food. divided by MLSS, mixed liquid suspended solids. So whatever is in suspended form, that is bacteria. Whatever is in suspended form in the aeration tank, that is a bacteria. So that we call it MLSS. Okay, suspended solids. So suspended solids are there. So food to microorganism, that is kg BOD divided by kg MLSS. That will give you food to biomass. So that is around 0.2 to 0.4 we have to maintain. Always we have to maintain 0.2 to 0.4. And MLSS we have to maintain to around 1500 to 3000 milligram per liter, means around 2000 milligram per liter. We have to maintain the MLSS, that is mixed liquid suspended solids. Okay. So you know, for maintaining this in the reactor 2000 or 3000, we have to recycle. So recycle ratio for maintaining this is 0.25 to 0.5. What does it mean? QR by Q. What does it mean? Whatever the waste which is going out of aeration tank, 50% if you recycle, if 50% recycle, then it is 0.5. If you recycle 25%, it is 0.25. So this much we have to recycle. Generally, we recycle around 40 to 50% of the whatever the sludge which is going out. We recycle that 40 to 50% of that. Okay. Now, for what is the hydraulic retention time in aeration tank? Means for what time our wastewater will be there in the aeration tank. What is the reaction time for that? So that is 4 to 8 hours. Means our wastewater will aerate in the reactor for 4 to 8 hours. Okay. For 4 to 8 hours it will be remaining in the reactor, then it will come out and go to the secondary sedimentation tank. Okay. So 4 to 8 hours that reaction time is there in the reactor. So what is this mean cell residence time? This is also called SRT, sludge retention time. It is 5 to 15 days. So what does it mean? Why the sludge retention time is in days and the wastewater retention time, that is hydraulic retention time, is in hours, 4 to 8 hours? Why this like this? This is 5 to 15 days and this is 4 to 8 hours. Why there is a difference in this? Anyone knows the difference? Why the sludge retention time is more, or mean cell residence is more than the hydraulic retention time? I have given you the difference, that hydraulic retention time, for what time our wastewater will remain in the reactor. That is hydraulic. And sludge time retention time is the retention time of the sludge. So why it is more? Anybody who knows? Okay, because why it is more? Because we are recycling the sludge. Because biomass is coming again and again in the aeration tank. Biomass means sludge is coming again and again, so its retention time is more. But our wastewater is coming once and it is going after 4 to 8 hours. Okay, so that's why the hydraulic retention time is 4 to 8 hours and the sludge retention time is 5 to 15 days. Because we are recycling the sludge, not the wastewater. Okay, so that is how. So there are different type of processes. This modified processes are there. Activated sludge process is modified into tapered aeration. Tapered aeration means we are giving the aeration first more, then second less, then third. Wherever it is required less, we are providing less oxygen. Wherever the air is required more, we are giving the more air, like that. Step aeration means that we are after every step, we are after some distance we are giving the aeration. then there is a completely mixed also. Then high rate aeration means when we are putting more air. Pure oxygen means what we are doing here, instead of air we are giving the pure oxygen only. Okay, it is a costlier, but that is also one system, and it has to be somewhat closed also, not very open, because all the oxygen that we give, that will go out. We don't have oxygen for our corona patients and like that, and we are giving pure oxygen to that. So that is not affordable also. That is going in some countries they are doing it, or some very less for less wastewater we can do it, but not practically feasible for a very large wastewater treatment plant, this pure oxygen type. And then comes the extended aeration. Extended aeration system is the system of the activated sludge where we give the hydraulic retention time is around 20 to 24 hours. Means your wastewater will remain there for 20 to 24 hours, not 4 to 8 hours. So what will be happening in extended aeration? That one thing is that your food by microorganism ratio is very less, because food will become very less in 20 to 24 hours, because the microbes will use it in 20 to 24 hours. So this food by microorganism becomes less, because the food becomes very less. Okay, microorganism is reaching up to 3000 to 6000. MLSs will become 3000 to 6000. Here you see MLSs is 1500 to 3000, and here the MLSs is 3000 to 6000. Why? Because it is there for 20 hours to 24 hours. So the bacteria will increase. It will consume all the organic matter, but the food will decrease. So the food by microorganism ratio is 0.05 to 0.15. Here the food by microorganism is 0.2 to 0.4. So this extended aeration is generally used when we are going for nitrifications also, or other thing that when we want very less sludge. Okay, like in our you see packed type of reactor. Okay, package reactor we say. Like if we have a very small colony or a building, multi-storied building is there. So there we go for extended aeration. Why? Because the sludge which is coming back, here we go for, you see here, what is the QR by Q ratio? Around 0.75 to 1.5, means whole sludge we are recycling. Whole sludge we are recycling, then the sludge problem will not be there. Here, if you see, whatever the this sludge is there, the waste activated sludge, you can see here, some sludge is recycled, and all other waste activated sludge that we have to manage it, either by sludge digestion or some other methods we have to manage this sludge. So this waste sludge will be generated. But in extended aeration, what we are doing, we are recycling the total sludge we are recycling, so no waste activated sludge is generated. Okay, so but if we are recycling total sludge, the air requirement will be very much high. If air requirement is high, so that we can do for a very small treatment, like for multi-storied building we can do it. Okay, why? Because we don't want this sludge. Otherwise we have to give the treatment to this sludge. So that in that system we do this extended aeration. So this extended aeration is done in the packaged, means very small treatment system. If it is there, we can use this process. Another thing is the attached growth process. So up to this, these are all suspended growth process, means bacteria is suspended in the reactor. Bacteria is there, you can see here in the photograph, this brown color that you are seeing, that is the bacterial color. Okay, that is called activated sludge. So bacteria is suspended in your wastewater, and it is cleaning your wastewater. But what is there in the trickling filter or attached growth system? that this, you see here the bed of plastic media is there, bed of media, and the wastewater is coming from upwards and it is going like this. Okay, so waste, this is the rotating arm is there. So wastewater is coming here. I can show you here. You see the wastewater is coming here, going up, and then it is trickling from above, and here the media is there. Nowadays the plastic media, earlier it was the rock media was there. So what is happening on the plastic media, the bacteria is not suspended, it is attached to the plastic or the rock. Okay, it is attached to that. You see, I can show you. Okay, first we'll see here. Let us say this is the, that media is there, or plastic media is there, and you see the bacteria is attached to this media, and wastewater is coming from upwards. So whatever the bacteria which is attached to the media, same reaction will be happening. Organic matter plus oxygen gives CO2 and H2 and other oxidized products. Okay, so your wastewater is treated. Okay, because organic matter is there, it is converted to CO2, carbon dioxide. Your wastewater is treated. So here only the difference in that suspended and this trickling filter or attached growth type system is that here the bacteria are attached to the media. Okay. So it is like this. You see this type of media is there, plastic media. So bacteria is attached here on the surface of this. More surface area of the media, more bacteria will attach. So we are designing the plastic media in such a way, this plastic media is designed in such a way that it will having a more surface area, so that the bacteria will attach, and we want the void percentage also more, because the wastewater has to go down. It should not stop in between. So the wastewater is going down also, the bacteria is treating also, treatment is done, and then the treated water is going downwards, like you can see here. So you see the media is here, wastewater is coming down. Whatever the bacteria which is attached to this media, they will consume this organic matter, convert to CO2 and H2O, and then it will come down, and from here it will be going to the clarifier, that is the sedimentation tank. In sedimentation tank, the sludge will be separating. We separate the sludge like this, and the treated water will be taken out. So this is the same. Only thing in this trickling filter is that we have to recirculate or recycle or recirculate, not the sludge. Here we are not circulating this. So that is the, in our students also, they are making mistake. What they are sometimes think that the sludge is recirculated. The sludge is not recirculated here. Here the wastewater is recirculated again. Why it is recirculated again? Because to have getting the more BOD removal efficiency, because first time let us say 70% or 60% will be there, second time again it will go, and again it is 60%, so the BOD will reduce and reduce. So 1 or 2 times recirculation is done in the, so this is the table. So there are low rate filters, there are high rate filters. This depending upon hydraulic loading. Here we are concerned about the hydraulic. Here we are not concerned about the F by M ratio, food by microorganism ratio. We are not concerned here. Here we are concerned about the hydraulic loading and organic loading. Organic loading is what? How much kg BOD per meter cube of the reactor per day? How much kg BOD we are giving per meter cube of the reactor per day? So around 0.5 to 1 kg BOD per meter cube per day in high rate filter. High rate filter is made up of plastic media. Low rate filter is made up of generally the rock type of filter. Here we are recirculation is done in high rate filter. Okay. Here because here no recirculation because it is so slow. Low rate filter is so slow that all this degradation will be done as it comes down. But here we have to do the recirculation. But this is very fast. High rate filter, they are very fast. nowadays we are having all the tower filters are there, because it is having a, I think, 3-story building. Tower filters are there because this 3-story building, the media is filled up and the wastewater is coming from upwards, and then this doing like degradation is done. Next is sequencing batch reactor. So after sequencing batch reactor is also a conventional active, but it is also activated. Everything is doing same activated sludge process system, but everything is happening in a one tank. There is no separate tank for reaction and for settling. Here you have seen the 2 separate tanks are there. One is aeration tank and one is secondary clear tank. But in SBR, that is sequencing batch reactor, everything we are doing in one tank only. You see how it is done. So how it is done that first of all, influent is coming in the reactor. Like let us say one reactor is there. This is the same reactor, only the phases are different. Only one reactor is there. So let us say wastewater is coming here. So that is called a fill phase. Okay, filling is done. After filling, we stop the filling and then we do a aeration or mixing. Okay. So same thing, the reaction, same reaction is happening here. What is the reaction? Organic matter plus oxygen gives CO2 plus H2. That reaction is happening here. So in that reaction, all the BOD, that is organic matter, that is reduced from, let us say, 500 milligram per liter to 20 to 30 milligram per liter in the react phase. So here we are doing the aeration. Okay. Here we are doing the aeration in the react phase. And then what we are doing, that after react phase, the whole sludge, the sludge will be settled at bottom. Okay, the sludge is settled, and then we draw the upper portion. Okay, then settling will be there, and take the upper portion, we decant it, and then the sludge will be remaining here. Then again we fill it. So what is happening here? Simply, this phase is of settling tank. What is done? This phase is of aeration tank. So in same, we stop the aeration. After react, let us say 4 to 8 hours we react. After that we stop the aeration. So what will happen? The sludge will settle. Whatever the bacteria is settled here. And then what is the supernatant? Supernatant is the treated water. It is free of organic matter. Why? The organic matter is already consumed here. So this is free of organic matter. That is treated water. We take out the treated water in the decant phase. All treated water will be taken out. What is there at the bottom? Bacteria, biomass. Then there is the idle phase. Then again with fresh water, fresh wastewater will come. Then again react will be there. Then after reaction is completed, we settle it. The treated water will be removed here. Again same process. So this is called sequencing batch reactor. Means in one reactor we are doing all the processes of the degradation and settling. Everything is done. So sequencing batch reactor is normally nowadays very much in use. Many effluent treatment plant, like I have seen in Mumbai, I have seen in Surat also, many effluent treatment plant, they are now operating on this principle, SBR, sequencing batch reactor. The one other advantage of using this is you can do the nitrification and denitrification also possible in this, because we can control the amount of time it will be remaining there. So nitrification is what? Okay, first I tell you, nitrification is what? Means Generally in activated sludge process, what we are removing the carbonaceous organic matter. Let us say example of a glucose, C6H12O6, or a sugar is there, C12H22O11. So these are all these type of things are removed in the, but if like protein is there, it is also degraded and coming to, let us say, NH3 or NH4. But that is not removed. Nitrogen is not removed. It converted into the form of ammonium or ammonia, but it is not removed totally from that. So for that purpose, we have to give more oxygen. So what will happen? The nitrogenous bacteria will convert the ammonia NH3 to NO2 or NO3. Okay, due to the oxidation, this nitrogenous nitrification or nitrifying bacteria will convert the NH3, and you know the reaction is like this: NH3 plus O2 in presence of oxygen, it will convert to NO2 and NO3, nitrites and nitrates. Okay, it will convert. Then comes the denitrification, right? Denitrifying bacteria will convert this NO2 and NO3 to N2 plus O2. Denitrifying bacteria which do not require oxygen, that is in anoxic condition, it will convert NO2 and NO3 to N2 plus O2. N2 is a gas, nitrogen gas, oxygen is also gas. So it means your whole nitrogen is removed from the system. So that is called nitrification and denitrification. In nitrification and denitrification, what we do, we give more oxygen after the carbonaceous organic removal. We give more oxygen so that all this converted to all the this nitrogen that will be converted to NO2 and NO3, and then after it convert to NO3, by the denitrification we are converting into nitrogen and oxygen. So that is called denitrification. So that is called nutrient removal. This nutrient removal technique is this. Nitrification and denitrification is the nutrient removal technique, okay, in which we are removing the nitrogen and phosphorus, because that is also important. In India, we are not doing, earlier we were not doing in our, what we are doing, we are carbonaceous BOD we are removing, and then we are just dumping into the river. So what will be happening in the river due to this nitrogen and phosphorus, the nutrients, what will happening, that algae growth will be becoming in the river, and you know, you have seen the eutrophication in the river. So eutrophication will be there in the river due to this nitrogen and phosphorus. So nowadays, due to the technology, and there we are using this SBR technology, that is another, I think another subject. So we are not going into the detail of nitrification, denitrification, because biological nitrification, denitrification, in that only thing you should know that SBR is used. How it is used, that is a separate topic. So what is the advantage of this? That it requires a small space. SBR requires small space. It is controllable react time, and that I was told. We can control the time. Okay, react. How much time you have to react, you can control it. And there is a settling, high nutrient removal capability. So I have told you just now, how high nutrient means you can remove the nitrogen also or phosphorus also by using this technology. BOD removal efficiency is also 85 to 90%, and there is no filamentous bacterial growth in this type of reactors. Limitation is that higher level of sophistication is required because timings control will be there. Maintenance is also very important because there is a sophisticated control, automated valves will be there, because you have to automatically switch off the aeration, then you have to start the aeration, then you have to take all the supernatant out of the system, then you have to fill the system. So everything is automated here. So all the devices, to run the devices and everything, you should require a very high level of sophistication in SBR. Generally in activated sludge system, that type of sophistication is not required. Next is the rotating biological conductor, RBC. What is this? This is also same as the trickling is that attached growth process. Means the bacteria is attached to the disc. You can see here the disc are there. This is the disc, and this disc is made of plastic media and it is rotating. Okay, this disc will provide a large surface area for bacteria to. So bacteria will be attached to the disc, and the water it is submerged. It is submerged like 40% submerged, and 60% will be above the air, in the air, and it is rotating. It is rotating. So 40% is submerged, rotating around 1 to 2 revolution per minute. It is rotating, one means in 1 minute it is rotating 1 revolution. So what happened due to rotating? What is happening due to rotating, you know? As it is rotating, the bacteria will be coming in contact with the food, that is organic matter at the bottom, because wastewater is there at the bottom. So bacteria which is attached to this this drum will come in contact with this, and then when it come outside, it come in contact with oxygen. So it is getting both the things. It is getting the food, that is the organic matter, and it is getting oxygen also, and it is rotating like this. Okay, and same reaction is happening. Organic matter plus oxygen gives CO2 plus H2O, and everything is treated. All the organic matter is removed. All the BOD is removed. Organic matter means BOD. BOD is removed. Okay. So advantage is RBC process, rotating biological contract is simple operation, low energy requirements. Nitrification is possible in this, and wide flow range, large biological population can be maintained on the disc, and it can handle the shock load also. So this is the disadvantage, like it has a limited controls, enclosure, limited experience and training is there. Now comes the RBC components. So the components of this is, you see, there's a contactor, that is where on which the discs are arranged, plastic discs are there. There's a tank in which the effluent is coming, and then after this everything is going into clarifier. Why a clarifier is needed? Because we want to separate the biomass from the treated water. Clarifier is required after every biological treatment, because we want to separate the treated water from the bacteria biomass. The biomass is settled down and the treated water will be taken out. So this is you can know, and this drive systems will be there. You know, the enclosures are to be provided like this. So you see here, this is the picture of that. It will be rotating like this, and on this plastic media, bacteria will be attached to this plastic media. The actual picture of the rotating biological contactor. Now it has to be covered also. Why it has to be covered? You know, why the cover is required? Because if you don't cover, there will be a algal growth. You see the cover is there, you can go inside this. You see the cover is there and you can go inside the rotating biological contactor, it is moving. You see so many. If you don't cover it, then algal growth will be there. Okay, we don't want we want only bacterial growth. We don't want the algae to grow, because if it is not covered, sunlight will be provided. If the sunlight will be provided, the algae will be growing on it, because algae, you know, that is capable of photosynthesis, and in the sunlight, if it gets the nutrient, it will go for photosynthesis. So algal growth will, we don't want this algal growth to be there. So this is the building is there, permanent building is there inside this RBC is there moving. Many this is the one case study where earlier activated sludge process was there, and there it is not working properly because of increased wastewater flow rate, inefficient pre-settling, final sedimentation tank, and not optimized sludge reflux. Then what they do, they provided this rotating biological contactor. The company has and after that they are mixing the sanitary wastewater 75 meter cube per day with their industrial wastewater 30 meter cube per day, and then two rotating biological contractors were provided, and then it is giving very good efficiency of BOD removal efficiency. Now next is the membrane bioreactor. I think membrane bioreactor, there is a separate lecture also on this. Therefore I will not go much in detail in this membrane bioreactor because one separate lecture is on this. Only something I will tell you that in membrane bioreactor we are using the membrane for this purpose, settling purpose. What is the need of settling? Settling need is to remove the biomass from the treated water. This clarifier, what is the need of this clarifier after biological? To remove the biomass from the treated water. So that is done by in the membrane bioreactor, that is done by the membrane filters which are there. So this membrane filters, either they will be inside the reactor, either they will be inside the reactor or they can be in the separate reactor. Okay. So, and the wastewater which we are getting after this membrane bioreactor is very good quality. Turbidity less than 0.2 and NTU. BOD you can go up to less than 2 milligram per liter. TS is less than 2 milligram per liter, nitrogen less than 3 milligram per liter. So very good quality. This is the normal effluent quality, and this you know after this MBR effluent quality. There are other benefits like better control of biological process, and 100% TSS removal will be there. We can use this water. Okay, so wherever the reuse of water is required, there we can use this MBR systems. So you can see here a membrane MBR process. What we are doing, we are removing this after activated sludge directly. This membrane process is there. We don't need this all things secondary clarifier and everything, or primary clarifier also we don't generally. So membrane processes. If we want to have a tertiary treatment, we can have one other reverse osmosis process after this membrane process. If we want to reuse it for better for reuse of water in industries or everything, we can have a RO system also where the less TDS is required. Otherwise, we can go for this only this UF membrane system. So I think I will not go in much detail because a separate lecture is there. This basis. Only this thing I will show you that this is the feed is there, and here the whole reaction will be happening. After reaction is completed, the wastewater is passing through this membrane. So when it is passing through this membrane, the whatever the suspended solids, they cannot pass through it. So only the treated water with very less suspended solids will go outside of this system. So this is the submerged MBR systems. Next comes the anaerobic reactors. So this is whatever I have told you, that is about the aerobic reactors. Now next comes the anaerobic. Now earlier, whatever the anaerobic reactors like we have used, it is the normally very low rate anaerobic reactors, but by the very much highly research, there are so many different types of anaerobic reactors that are created. As you know, anaerobic reaction does not require oxygen. Okay? It does not require oxygen. So all the organic matter is converted to carbon dioxide, that is 20 to 30%. Methane is around 60% you will get, and water. So CO2 plus CH4, that is methane, plus water you will get. So very high concentration of active biomass in the reactor unit. And other thing I want to tell you, the biomass yield ratio of the anaerobic reactor is very less. Biomass yield ratio means how much biomass is generated per gram of BOD or organic matter. That is very less here. So therefore, why we are using it for sludge digestion? You know, after activated sludge process, whatever the waste activated sludge is there, we are removing it in anaerobic sludge digesters. So what is sludge digester? Sludge digester is nothing but an anaerobic treatment, and due to the less biomass production in this treatment, everything is converted into methane and carbon dioxide. So you will get very less sludge. So sludge management will become very less if you use the sludge digesters after. So all the, if you have seen in your, if you have visited the sewage treatment plant, all the sludge which is generated from activated sludge process, they are going into the anaerobic sludge digester, where the due to anaerobic reaction, the sludge quantity will become very less. Okay. Sludge is what? Biomass is what? It is also organic. Everything is organic, you know. Biomass is also organic. Do you know what is the formula of one bacteria? Anyone knows the formula of bacteria? Chemical formula of bacteria. Chemical formula of bacteria. That is C5H7NO2. Okay, this is the chemical formula of bacteria. Okay, so bacteria is also an organic matter. So whatever the sludge is generated, that is organic matter. It is the food for anaerobic bacteria. It will be used by the anaerobic bacteria as a food, and it will be converted into methane and carbon dioxide. Okay, so we are also organic matter. Like bacteria is organic matter, we are also organic matter. Our body is made up of organic. Okay, we are also organic matter. But we cannot give a formula to us. Can you give the formula of your body? Your body. What is the formula of your body? Chemical formula. Anyone knows the chemical formula of your body? Like I have told, the bacteria has the chemical formula C5H7NO2. Anybody knows the chemical formula of their body? No, we cannot, because we are multicellular organism. So many cells are there. We know the bacterial because it is a unicellular organism. We can find out the this may be from here on there, C5H7NO2, something more or less. But for our body, we cannot tell because we are multicellular. So many cells are there. How many cells we don't know. Okay, so we cannot give the chemical formula for our body. But for bacteria, that is unicellular, we can give. So yes. So what I want to say is that that sludge, which is composed of biomass, bacterial sludge, bacteria, anaerobic bacteria, which is it may be dead or it can be live. Okay, so that is whole thing is digested by in the anaerobic reactor. Can be anaerobic. But nowadays, high rate anaerobic reactors are there, which can treat the high COD or high BOD wastewater also, which can treat directly. You can feed into the anaerobic reactor, and you will get the good COD removal. And that is very much relevant to the high BOD COD wastewaters. Like, you know, in distilleries, because I have done some work on distillery, many other food waste also, or many other industries also, they have very high BOD and COD. Do you know how much COD and BOD is there of distilleries or sugar industry wastewater like that? Anyone knowing?
So COD will be around more than 1 lakhs.
Yes, it is around 1 lakh milligram per liter. BOD is around 60,000, 70,000 milligram per liter. You know, what is the BOD of our sewage wastewater? That is domestic wastewater, which is generated from our house. What is the BOD of that? Anyone? What is the BOD of the wastewater which is generated from our house or sewage? That is around 300 to 400 milligram per liter. You see the difference. We are very worried that 300 to 400 milligram per liter, if it goes to water bodies, what will happen to the aquatic life? And here we are seeing the industries which are having 70,000 milligram per liter BOD. If you directly discharge into the river or any water bodies, what will happen to the aquatic life there? In no time the DO will become zero if you discharge that type of wastewater. Okay, so when the BOD is very high, this type of anaerobic treatment is very much useful. Why? Okay, this type of high BOD, if you say, sir, why anaerobic, can we not give directly to aerobic treatment? What is the problem if we give directly to the aerobic treatment this high BOD wastewater? Anyone knows what will happen if we directly feed high BOD wastewater to aerobic treatment, which is not a feasible solution? We generally give to the anaerobic treatment first.
Microorganism and the nutrient load and this thing, your aeration load will be very high then in that case.
Yes. You know, organic, I have told you in the initial that organic matter, if it is more, the bacteria will start degrading it and it require more oxygen also. So I am talking about 300 to 400 milligram per liter. And if you have a 40,000, 50,000 milligram per liter of BOD, that is so much high organic load, the bacteria will start consuming, and if you have to survive the bacteria of aerobic bacteria, you have to give oxygen, air, so much air you have to give. And then air compressors, like yes, so Chitra Rajput Chauhan, she has written, with very high electric consumption will be there. The compressors, you see how much air you have to provide, and your whole जितना production नहीं होगा उतना तो treatment plant में चला जाएगा. You know, mainly why the people are very much afraid of running the ETP or STP in the industries, because their electricity bill is very much high in the STP. No industry people will if there is no law, environmental law, they will not run the ETP. No industry, because they think that is the waste. कचरे में पैसा क्यों डालना? Okay, why to make a waste this much electricity in treating the wastewater? Okay, so some industries are doing that. They are doing some theft. They are not running their ETP or not running the air compressor. And when this people sometimes comes, pollution control board, they catch it. Okay, many industries they are doing, and then there's so many things come in play. You know, there are so many corruptions and everything comes in play. So this is the thing. So in that type of effluent, what we have to do, we have to use this anaerobic treatment. Why? No oxygen is required, no air is required. Only what you have to do, just cover this reactor, mix it properly, and you will be give some input like some anaerobic bacteria to start it, and then you monitor it properly. Why we have to monitor it? Because anaerobic reaction has two phases. Okay, anaerobic reaction has two phases. First phase is acidic phase, where all this organic matter is converted to acetic acid, propionic acid, and other acids. So that is called acidogenesis. And after that, these acids will be converted to methane. That is called methanogenesis. So two phases are there in anaerobic. First, all the organic matter is converted to acids like acetic acid or propionic acid and other acids, and then that is called acid formers or acidogenesis. And after this acids by methanogenesis or methanogenic bacteria, that is converted to methane, CH4. So these two processes has to be these two processes are there. Okay, so there you have to see in anaerobic treatment, you have to see that and you know carbon dioxide is generated and it will be there in the system because it is closed. So it is dissolved also in the system. So CO2, when it is dissolved, it forms the carbonic acid and the pH becomes less. And if pH becomes less in your anaerobic reactor, your whole microorganism will die. Okay, so what you have to do? You have to add some alkaline material. Alkalinity has to be added. Okay, if your pH base, so that is to everything has to be monitored. pH you have to monitor, because if the pH will become less due to the carbon dioxide and everything, then you have to add the alkalinity in form of some caustic soda or something else. But if you are fortunate, this is happening generally in the sugar waste where there is no nitrogen is there. If you have a protein or nitrogen in your wastewater, what will happen? Ammonia is generated, and you know all the chemistry people are there. When the ammonia will be generated, then your pH will increase. So that will be balancing your pH. Whenever it is converted to ammonia, and then the balancing will be there. But if the ammonia also become very much high, then also it is a problem. Then the ammonium toxicity will be there and your whole pathogenic bacteria will die. So that is also not very good if more ammonia will be generated. Okay, so that balance has to be. Therefore, anaerobic treatment is very much sophisticated in that way that you have to control it in very good manner. You have to always see that what is your acid by alkalinity ratio, that is called BFA by alkalinity ratio, volatile fatty acids by alkalinity ratio, that you have to always monitor. You have to monitor the alkalinity also in your reactors. Yes.
Shanti Hospital, madam.
I think some okay. So okay. So that has to be our this has to be. Now there are so many anaerobic reactors, anaerobic contact process, anaerobic filters, USB, very frequently used technology everywhere. USB technology, flow anaerobic sludge. So we'll see one by one that in anaerobic contact process, there is a complete mixed reactor is there, and biogas is generated from here, and it is same as activated sludge process but it is an anaerobic activated sludge process. Okay. So that is called anaerobic contact process. Then there is an anaerobic filter. Anaerobic filter is like this. You can see here. This is the filter media is there, and you know feeding is done. Let us say from here, feeding is done, and through the media where the anaerobic bacteria are there, this degradation, anaerobic degradation will be there, and then from the top the effluent is taken out, and whatever the biogas, that is the methane, is generated here, that is also taken out. So that is a upflow anaerobic filter. So there are two types. There is a upflow anaerobic filter and there is a downflow anaerobic filter also. Now the media which is will be used in anaerobic filter is, you can see here, earlier rocks were used like in trickling filter, but nowadays plastic media is used, having the void volume of 85 to 95%. If you don't provide enough void volume, there is a chances of blockage of this or clogging of this. Then there is a downflow anaerobic filter also. After that, whatever this USB process, that is upflow anaerobic blanket process, very much nowadays it is used. It is developed in 1970 by Lettinga in Netherlands, and what it is, the process is that in this process, this anaerobic bacteria will form a granulation. Granules are formed of 1 to 3 mm diameter. So as the granules are bigger in size, they will settle down and retain within the reactor. Means they will not go outside the reactor. Therefore, they will treat the effluent very fast. Very low HRT here. I've written 4 hours, but it normally 6 to 8 hours. It will be there, and it will be converting your, just like aerobic, it will be converting your BOD and treating the effluent. So USB is very much nowadays in use in industries where high BOD and COD is there. Like you see in every distilleries you go, first unit is USB reactor. So how it is there? You see the effluent is coming, going upward, and this anaerobic bacteria will form a granules like this, and these granules are somewhat heavy. You see the this orange color, they are the anaerobic bacteria or granules. Anaerobic bacteria granules are there, so they will be retained in very much high quantity, and therefore they are treating the waste in a very efficient manner. And on the top of this, you have a separator. You see the granules photo are there. This is the anaerobic sludge granules. This type of 0.5 to 2 mm size, these granules are there, which is formed inside. And there is a gas liquid separator above. So the gas will be coming outside from like this, and the liquid will be coming outside, and your effluent, treated effluent, will be coming out like this. So the biogas like methane and CO2 will be coming out. Another advantages of using this anaerobic bacteria is you are getting the methane. Methane is very good biogas which can be used for as a fuel. Okay. So that is also advantage of using having this anaerobic reactor. So USB reactor is, this is the design criteria. I have given temperature range and sludge retention time, and I am not going into much detail in this. Static granular bed reactor, other type of reactor, just opposite to USB from the above, flow from top to bottom. Now we have to be very careful in the anaerobic system, because if the sulfates are there, so these sulfates will be converted to the sulfide by the sulfate reducing bacteria, and this sulfate reducing bacteria will compete with the methogenesis bacteria, and it will stop your anaerobic treatment. So these hydrogen sulfides are produced, which will be, that is called sulfide toxicity. So you have to be very careful if in your wastewater sulfates are there. You have to reduce the sulfates first, and then you have to do this. Otherwise, 50 to 250 milligram per liter, if the sulfides are there, they are very much toxic to methanogens. Then there is expanded bed reactor where the biocarriers are there. And then the fluidized bed reactor where the similar to FBR, but bed expansion is 25 to 300% in this. Then you can have a hybrid system, means USB with anaerobic filter. And this anaerobic baffled reactor, like the this is the wastewater is going like this, and there is a baffle here. So, and you see the sludge is there at the bottom. So the waste is going like this. This is very simple system we can have. So I think I will stop here, and if any questions are there, we will discuss some of the questions. So I have seen in the way some questions are there. like what is the composition of sludge that Suchitra Rajput Chauhan has? So sludge is nothing but biomass, that is bacteria. So that is that I have already told that it is a bacteria. Yes. Any other question if you want, you can ask. You can write in the chat section also. I know this is the last session. We are feeling tired also. Okay, one question is it advisable to have this treatment after photocatalysis in case of textile diabetes? Okay, it is depends upon what is the BOD by COD ratio after this treatment. Okay. So what you have to do, you have to find out the BOD, you have to find out the COD. BOD gives you the biodegradable organic matter, COD gives you all the organic matter. If BOD by COD ratio is less than 0.5 or less than 0.3 or 0.2, you cannot use this biological treatment. The biological treatment only you can use if your BOD by COD ratio is around more than 0.5, like 0.7, 0.8, like that. So you have to first find out what is that ratio.
Sir, one question: is there any rise in temperature, sir, during this process?
Temperature, yes, temperature should be around 25 to 35 degrees Celsius. Generally, all the systems. In anaerobic system, one other temperature, that is thermophilic digestion, is there. That can be happening at around 45 to 60 degrees Celsius, also up to that. But generally, whatever the ambient temperature we have, 25 to 35 degrees Celsius, this process is happening. Other thing is pH. pH has to be also near neutral, like around 6 to 8 pH. This is a biological system. So if your pH will be less, all the bacteria will die. If pH will be more, the bacteria will die. Everything is dependent upon how the bacteria works there.
Okay, thank you, sir.
Okay, participants, if there is no question, we will be yes. Here, okay. So we will be finishing here and there. Thank you. I think now we will be ending the session, and tomorrow we will be meeting at 10:30 for tomorrow's session. Okay, thank you.
Thank you, sir, for delivering wonderful lecture. And for all the participants, I have arranged tomorrow's session also, link tomorrow lecture also. So again, same process you have to follow. Go to the channel. Thank you very much, all the participants. And now, if you want to leave, you can leave. You can end our session.